Now we come to the second part of our physiology lectures of the second week. We will continue the process of the white blood cell genesis and function. Now we come to the process of phagocytosis. Phagocytosis refers to the process of cellular ingestion of the offending agents، يعني طريقة اللي cells تقدر بيها تقضي على offending organisms whether bacteria or debris or whatever by ingesting them. So its most important function is mainly performed by, as we said early in the first part of our lecture, it's mainly formed by neutrophils and macrophages. We have some differences between the phagocytosis performed by neutrophils and the phagocytosis performed by the macrophages. Now, the neutrophils are already active when they enter the tissue. They are released in the blood, they are active, and when they move from the blood to the tissue, they are already active. A single neutrophil can usually phagocytose from 3 to 20 bacteria before they become inactivated and die. Now, how this process of phagocytosis takes place? When the neutrophils comes in contact with the cell, now it will surround it by extending pseudopodia surrounding the cell, and then the pseudopodia are going to form a closed chamber around the particle, and this is called the phagosome. And then the lysosomes or the digestive granules inside the neutrophils or the macrophages are going to attach to the wall of the phagosome, and they are going to deliver their granules inside the phagosome, and then those granules are going to digest whatever present inside the phagosome to form what we call a digestive vacuole. And then the residual contents are extruded outside from the cell. The neutrophil, after performing this function, are usually going to become inactive and die. While for the macrophages are much more powerful than the neutrophils. They are capable of destroying as much as 100 bacteria and even larger particles than the neutrophils. Furthermore, after the digestion of the particles, the macrophages can extrude the residual products and survive and perform their function for many more months. So they are more potent and they can live for a longer time, performing more and more function than the neutrophil. But the basic process, طريقة اللي the two cells performing their function is usually identical. As we say, when they come in contact with the cell, they will extend pseudopodia للأذرع surrounding the particle, and then they close, forming a chamber we call the phagosome with a floating particle inside it. بعدين تجي lysosomes and the digestive particles and the granules, they come in contact with the wall of the phagosome, deliver their content inside it, and they will digest whatever inside to become a digestive vacuoles, and then extrude their content and eliminate them outside the body by the process of exocytosis. So the killing after phagocytosis is done how? By, as we said, the lysosomal contents of the cytoplasmic granules, when they release their content inside the phagosomes, this is where it becomes what we call a digestive vesicle. We have other methods, which is in far also potent when the process of phagocytosis sometimes fails because the bacteria, for example, has a very thick wall or whatever other factor that prevent the process of phagocytosis. شلون بهيجي حالة السيل راح تتصرف؟ شلون راح تقدر تقضي على offending agent? It can also kill most of the bacteria when the lysosomal enzymes fail, How? By producing powerful oxidizing agents اللي هي the oxygen free radicals like the superoxide, hydrogen peroxide, the hydroxyl ions. These are very powerful agents that are produced from the bacteria, from the white blood cells. Mainly they are being produced by special organelle known as the peroxisomes. Those reagents, or sometimes even by enzymes in the membrane of the phagosomes, whatever the process they are produced, those reagents are very lethal to most of the bacteria, even in small quantities. So, نعيدها مرة ثانية. After being phagocytosed, the offending agent is killed either by what? By releasing of the lysosomal contents that are going to hydrolyze and digest the offending agents into the phagosome, converting it to what we call a digestive vesicle. Or the bactericidal agents can kill most of the bacteria when the process of lysosomal enzymes fail because of thick wall or whatever other factors. How? By producing a powerful oxidizing agents, which are the oxygen free radicals like the hydrogen peroxide, the superoxide, and the hydroxyl ions. These are produced either by the granules in the wall of the phagosome or by peroxisomes, which are special types of organelles present inside the white blood cells. These substances are very lethal. Even in small quantities, they can kill the offending agent. Now, we come to the role of the neutrophils and the macrophages in inflammation. When you have an injury, what's the first body response to that injury? What's the first type of cells that come into that injury? What's the second type of response? The third type of response? The fourth? شنو السيكونس اللي راح تصير عندي when you have an inflammatory process? When the tissues are injured by any mechanism, whether it's chemical injury, whether it's trauma, whether it's infection, I will have inflammation. مو صحيح؟ لا تتذكرون من السيل injury والinflammation اللي أخذتوها بال unit 3. Injury to the tissue by any mechanism can result in inflammation. Substances are released by the injured tissue, and they will lead to secondary changes on the surrounding and injured tissue, and the collection of all these tissue changes, we call it what? We call it inflammation. Now, suppose that we are having an infectious process in the skin or on whatever part in our body. What's the first line of defense against this infectious process? The first line of defense against this infectious process is, in fact, the tissue macrophages that are already present in that tissue. ليش? Because they are already present. هي أصلا موجودة. So what will happen is that those cells are going to enlarge, develop, becomes more active in order to counteract the infectious process. But we have to keep in mind that the number of those cells is not much. يعني عددها حيكون قليل. It's not much inside the tissue. So the first line of defense will be the already present tissue macrophages. منو يجي وراها? The second line of defense will be what? The neutrophils. The neutrophils that are already present in the blood, they are going to get outside from the blood to the site of tissue injury, and as we said, the process where they leave the blood vessel, we call it diapedesis, and they crawl by amoeboid movement, and they are attracted to the area of inflammation or injury by what? By chemotactic substances. When you have an inflammation, we have certain types of substances and changes that happens to the cells. First, the neutrophils are going to be close to the wall of the blood vessels. This process we call it margination. And then they are going to leave the wall through the small pores of the capillaries through a process we call it diapedesis. Then crawl by amoeboid movement, attracted to the area of focus of injury by chemotaxis. So this is margination and then diapedesis and chemotaxis. طبعا هاي المفروض انتو كلها تعرفوها من inflammation، محاضرات الانفلاميشن اللي أخذتوها بال unit 3. So we are not going to talk about the details, just the outlines. So the first line will be the tissue macrophages. The second line will be the neutrophils in the blood, getting out of the blood, passing through the small pore of the capillaries, moving towards the focus of inflammation attracted by chemotactic substances released from the site of inflammation. وتتذكرون قلنا chemotactic substances can be bacterial toxin, viral toxin, complement substances, debris, degenerated tissue, whatever. It's going to attract the white blood cells, the neutrophils, to the focus of inflammation. Now, what's the third line of defense? The third line of defense will be the macrophages invading the tissue. As we said, monocytes are already present inside the blood. They are going to move outside the blood, inside the tissue. They are going to enlarge, swell, become active, and called once again tissue macrophages. But this process takes time. That's why initially we have neutrophils predominant, and then after a few weeks, days to weeks, the macrophages dominate the phagocytic cells because they are greatly produced by the bone marrow. They produce new monocytes and they will get outside of the blood to become tissue macrophages. When you have, in the second line of defense, when you have a neutrophilia, you have to remember that the bone marrow already have a store of neutrophils inside it. So when the neutrophils starts to get outside of the blood vessels, reaching to the site of inflammation, the number of the neutrophils inside the blood is also going to increase 5 times to its normal level. How? By releasing what's already stored in the bone marrow from the neutrophils. Okay. Now, the fourth line of defense is increasing the production of neutrophils and monocytes by the bone marrow. How? By the influence of many of the growth factors. مثل ما قلنا تذكرون اللي هي driving the pluripotent and the multipotent stem cell pathway of differentiation. شنو اللي يحددها؟ The growth factors, the interleukins, the cytokines, and many of these interleukins and cytokines, they are product of inflammatory process, so they will guide the bone marrow to produce the type of cells that the body demands. نرجع نعيدها مرة ثانية. The sequence. The first line of defense will be the already present tissue macrophages, because they are already present. They will become active and develop within minutes, but we have to keep in mind that the number is not much. And then the second line of defense will be the neutrophils getting outside from the blood to the tissue by margination, diapedesis, and chemotaxis. And also the number of the neutrophils inside the blood is going to increase 4 times, 5 times its normal level by releasing what's already stored of neutrophils inside the bone marrow. The third line of defense will be what? The monocytes that are already present inside the peripheral blood. They are going to get outside of the blood vessel to the inflamed tissue, and they are going to become active and develop into macrophages, so that after days or weeks of the invasion of the offending agent, the macrophages are going to dominate the phagocytic activity. Finally, The fourth line of defense will be the bone marrow starting to produce more and more neutrophils and monocytes and respond to the growth factors that are going to guide the pathway of differentiation that the body requires. مثل لنا ببساطة monocytes getting from the blood vessel outside the blood vessel into the tissue. They swell, become enlarged, and the tissue macrophages performing its phagocytic function. قلنا المacrophage, it has a very powerful ability. It can phagocytose about 100 type of cells, and it can extrude the residual content of those cells and perform its function again and again with a lifespan approaching months. The neutrophils, they are a large number. They get outside, attracted to the tissue, performing its phagocytic function mainly against microbial organisms, and they have the ability to phagocytize from 5 to 20 organisms before they are becoming inactivated and die. Now we come to the other types of cells in our body, of the polymorphic nuclear cells, which are the eosinophils. Usually they don't constitute a big percentage of the total white blood cells in our body, usually not exceeding 2-4% of the whole blood leukocytes. If we compare them to the neutrophils, they are weak phagocytes in comparison to the neutrophils. But they have certain important functions that are unique for them. They are produced in large numbers when you have a parasitic infection. In fact, they are very important in parasitic infection. Why? Because they have special surface molecules that can help them attach to the parasites, and they have substances released from them that can kill the parasites. Some of these substances are hydrolytic enzymes and modified lysosomes. Some of them are highly reactive oxygen species, just like the neutrophils, that are also lethal to the parasites. And one of the important things that they release is some of the granules, they have a highly larvicidal polypeptide we call the major basic protein. That's very efficient in killing the larvae that have a thick coat or a wall that's usually resistant to most of the lysosomes and the granules of the neutrophils. But the major basic protein of the eosinophils is a very powerful substance that acts against the wall of the larva. So the eosinophils are very important in parasitic infection. They become increased during parasitic infection, and they have ability to kill those offending agents. Also, the eosinophils are known to be increased in certain conditions or allergic reactions, such as asthma, for example. They increase in the blood and they increase in the lung of tissues for patients with asthma. Patients with skin allergy, whatever the cause of the allergy, they have increase in the number of eosinophils in the blood. So they increase in two conditions: when you have parasitic infection and when you have allergic reactions. The last topic of our lecture today will be the basophils and the mast cells. Both types of cells, they also are produced for the bone marrow, for the basophils, like any polymorphic nuclear cells. The basophils are produced and mature in the bone marrow before they are being released to the blood, and they're found in a very small number in the blood. As you recall, the absolute number of them does not exceed 0.1 from the total YBC count. The mast cells, on the other hand, they originate from the stem cells in the bone marrow, and they complete the process of maturation inside the tissues. Both of those two types of cells characterized by having receptors on their surface for the IgE immunoglobulin. So they have receptors for the IgE immunoglobulin. When the IgE immunoglobulin attach to that receptor, it will cause destabilization of the membrane. And when the membrane becomes unstable, it will open, and the contents of those cells are going to be released. Some of these important substances that are being released are heparin, histamine, bradykinin. These are mainly responsible for the allergic reactions associated with the increase in the number of those two cells, because one of them are local, causing local vascular reactions, tissue reactions, resulting in the allergic manifestations such as itching, rhinorrhea, sneezing, bronchospasms seen in type 1 hypersensitivity reactions. Thank you.